Hello friends, my name is Ali and I will explain about Mendel's experiment and gamete formation. In my project I will I will say about that and how we did that. Then I will say that with the slide and video. So the first one, Mendel's experiment. Mendel's experiment. Who is the Mendel? Mendel is the father of genetics, and he introduced the genetics by using the garden peel and using the units of inheritance. So he did three principles. There are the first one, the principle of independent assortment, the principle of dominance, and the principle of segregation. And the second one, I will say about the pear plants. But what is the pear plants? The function of pear plants is pear plants produce 5 generations and they have many different types. Then have genus and that are independent. So structure of the pear plants, that is the petals and that is the sepals. So Mendel, in his experiment, Mendel did choose breeding lines. Exhibiting opposite feature of inheritance. Then he observed the offspring produced from this one. Then he just chose the one parent. Then Mendel demonstrated it by crossing. There is 3 crossings. 3. Monohybrid. Dihybrid and then trihybrid. So he did that with that crossings. Then he did it by steps, like steps of Roland transfer. Did you see? Firstly he did open flower and reveals the pollen, anthers and the tip of carpel, and then transfer pollen from anthers to carpel. Then fertilization occurs and the plant seeds and observes the offspring. Then Mendel identified 7 pairs of independent traits controlled by the genes. Then he did more things and then that on pairs. So that is the petals, but that is sepals. Did you see? With using that pair, he introduced the genetics and the inheritance of that one, and he used that. And he introduced the genetics by the humans and the plants or HC. Then I will show my slide. Mendel's experiment. Gregor Mendel was an Austrian monk who chose the basic rules of inheritance. Then from 1858 to 1866, he bred garden peas in his monastery garden and analyzed the offspring of these matings. Then how he did that? And he used the garden peas. Garden peas was a good choice of experiment, because by using that he did anything, and he did 4 functions, and that is the seed texture, round and wrinkled, seed color, grain is yellow, flower color, white and purple, growth, habit, tall is dwarf. That is the colors of that functions, like how he did that. Then and the 3 others that also varied in a qualitative rather than quantitative way. So how he did that? And he did that like the crossings, his peas are normally self-pollinated because the stamens and carpels are enclosed within the petals. By removing the stamens from unripe flowers, the Mendel used that and he did the experiment. And the Mendel could crush pollen from another variety on the crops when they ripened. Then he did with 3 crossings. Do you know? Monohybrid, dihybrid, and trihybrid. That crossings helped to Mendel to do an experiment. First cross. All the pears produced in the second and the first generation were round. Then all the pears of this F1 generation have an RR genotype. All the haploid sperm and the eggs produced by meiosis, by meiosis. Then all the zygotes received one R allele and one R allele. Because the R allele is dominant to the R allele. Then the phenotype of all seeds was round. Then, we studied in the terminology of genetics, allele and the dominant allele. That is that can be illness. Then I will say the table: P gametes and the P gametes wrinkle pan and round pan. If we multiply their RR and the P gametes and the P gametes round and wrinkle. Did you see? The next one, the second cross. Mendel then he allowed his hybrid pears to self-pollinate. The wrinkle trait which did not appear in his hybrid generation, and he repeated in 25% of the new crop of pears. Then, in the table, I will show the table.